Welcome everyone to our third session this morning. You are currently in web room number 3, and the session that we have up next is identifying genes for zebra mussel control. It's going to be presented by Dr. Daryl Gohl and Dr. Scott Ballantyne. A reminder, as with all of these sessions, to put your questions in the Q&A box at the bottom of your screen. And you can enter questions in that box at any time. I'll be monitoring that question and answer box for when we get to that portion of the presentation and feeding those to our presenters. So no need to hold on to those questions, just pop them into the Q&A box and we will get to them after the presentation has wrapped up. With that, I'm going to turn over the Zoom to Daryl and Scott to introduce yourselves. And we can get started.
Great, thank you, Corey. Hi, everyone. My name's Daryl Gohl, and I'm going to be presenting today together with Scott Ballantyne from the University of Wisconsin River Falls about a project that we've been doing to try to identify genes that could be useful for zebra mussel biocontrol. So just to give you an outline of what we'll be talking about today, first I'm going to tell you about work that we did previously sequencing the zebra mussel genome. And this is work that really laid the groundwork for this follow-on project to try to start to use that information to genetically manipulate zebra mussels. I'll tell you a little bit about the approach that we're using, which is a technique called RNA interference. And then I'll tell you a little bit about how we're taking the information in the genome and using that information to prioritize genetic targets for testing in this RNAi system. And then I'll hand things over to Scott, and he's going to tell you a little bit about the work that we've been doing over the summer in the MESRC Containment Lab, working on developing delivery methods for these RNAi constructs, and also developing phenotypic assays that will allow us to look at the effects of these constructs. And I'd also just like to introduce the team that's working on this project. So we've been working really closely with Dr. Mike McCartney. He's a zebra mussel and invasive species expert that was also one of the leaders of the Zebra Mussel Genome Project. So he's been continuing to contribute to this project and our work in MCL this summer. Scott, who you'll hear from later in the talk, and then also 2 scientists within the UMGC, Lindsay Gengelbach and Margaret Donovan, have done a ton of work both on the molecular biology of making these constructs, and then also in helping to kind of set up and run experiments in the MESRC Containment Lab with zebra mussels over the summer. So I lead a group called the UMGC Innovation Lab. So we primarily work on next-gen sequencing technologies, developing and applying genetic and genomic technologies to a wide variety of questions. And this is just sort of a list of the various things that we've been working on over the past 7 or 8 years or so. But one large project that we carried out was the sequencing of the zebra mussel genome. As I mentioned, this was done in collaboration with Dr. Mike McCartney, and at that time was with MESRC. This was actually a very large collaborative effort. You know, sequencing a large genome like this is a really big job. And it involved contributions not only from MESRC scientists and University of Minnesota Genomics Center scientists, but also a large team at the Minnesota Supercomputing Institute at the University of Minnesota, who helped with the assembly and annotation of the genome, as well as a number of external collaborators at the NIH, at University of Toronto, and at a company called Phase Genomics. And this was actually a previous MESRC funded project. And this information, you know, is now critical for the work that we're doing in this current project. So just to give you kind of a brief overview of what we did in sequencing the genome, we actually sequenced the mussel that was sequenced was actually from the Duluth Harbor. And to sequence the genome, we integrated a wide variety of different technologies that we have in the Genomics Center. So this used the instruments that are shown here, Illumina sequencing, PacBio sequencing. These are really cutting edge sequencing approaches. They are sort of called third generation sequencing approaches that incorporate very long sequencing reads that let us get a really contiguous and complete assembly of the genome. We also integrated a even newer technique called Hi-C that helped us to scaffold these reads onto large kind of chromosome assemblies. So by merging all of these techniques and the really fantastic resources that we have at the University of Minnesota Genomics Center, we're able to make the most contiguous and complete molluscan genome assembly reported to date. So we have a really high quality now reference genome for zebra mussels. In the context of this project, we also acquired some functional information. So we're able to dissect different tissues out of zebra mussels and look at gene expression in those tissues. And that helped us both to annotate the genome and sort of make the list, catalog the list of genes that are in the genome. But it also gave us information about those specific tissues. And I'll get back to that a little later in the talk. So this is sort of a visualization of the genome assembly. And we're really excited to see that we ended up getting 16 chromosome length scaffolds in this assembly, which matched what was known from previous karyotyping work that zebra mussels had 16 chromosomes. So we think we have a very good and very complete map of the zebra mussel genome. And when we reported this work in 2019, and this was the headline in the Star Tribune, it caused a little bit of consternation among the team because of some ambiguous wording. But this was really the goal of the project, to take this information in the genome and then try to start to apply it to identify genetic weaknesses and ways that we could potentially use this information for biocontrol efforts in the future. But in addition to needing that genetic information that we got in the genome, there was another large challenge in trying to genetically manipulate zebra mussels, and that's the fact that they don't grow and propagate robustly in the lab. And it's a bit ironic given how robustly they grow and propagate in the lakes and rivers throughout the state. But when we bring them into the lab, we can keep them alive for a period of time. And actually, we've had some mussels in the lab, I think since July or so, that are still going. But really, you run into a problem in getting them to spawn and reproduce in the lab. There is an ongoing project by Ben Minarek from the Minnesota Zoo trying to work on this and trying to actually propagate the mussels for a longer period of time. And that might open up some other avenues for genetic manipulations in the future. But for now, we're a bit constrained in what we can actually do. So given these constraints, we turned to a technique called RNA interference. This is a approach or a method that was initially discovered in nematode worms called C. elegans. And what the researchers found is that you could inject double-stranded RNA into C. elegans, and this would knock down the expression of the target gene that double-stranded RNA corresponded to. And importantly, these researchers also found that you could deliver these double-stranded RNAs by feeding the nematode worms bacteria that express this double-stranded RNA. This was a really important discovery. It was found that this generalized to a variety of other organisms, and the researchers who originally discovered this got the Nobel Prize for this discovery, because this approach can be applied very broadly to manipulate gene expression in a variety of organisms. So I'm showing some examples here from Drosophila, from planaria, importantly from oysters, so another bivalve where this RNA interference technique has been applied. And it's even been used in humans. So just a couple of years ago, the FDA announced the approval of the first RNA interference-based therapy in humans. So, this is a technique that has potential for very broad application, including bivalves, and we felt that it would be a good way of potentially manipulating gene expression in zebra mussels. RNAi is thought to have sort of evolved as a innate antiviral defense mechanism. So there typically aren't double-stranded RNAs floating around inside cells, but there are a number of RNA viruses that have double-stranded RNA genomes. And so it's thought that this machinery may have evolved to kind of protect against these double-stranded RNA virus infections. And there's a whole machinery inside the cell that recognizes double-stranded RNA, cuts it up into small pieces, uses those pieces to search for other homologous or other matching mRNAs in the cell, and then degrades or silences those mRNAs based on that homology to the double-stranded RNA sequence. So in essence, what we're doing in this project is basically trying to trick the zebra mussels into thinking that they, or into mounting this sort of antiviral response against their own genes. So to give you a little bit more detail or information about how we do this in practice, what we do is we take a small segment of a zebra mussel target gene. We put this into an expression plasmid that allows us to produce double-stranded RNAs. And then this plasmid is placed into a strain of bacteria that will actually express those double-stranded RNAs. And then the plan is to basically take these double-stranded RNA producing bacteria and feed them to the zebra mussels, and then to score the phenotypes that are induced. And we can either look at visible phenotypes, look at measure aspects of their physiology, like how they filter, or we can also look at molecular phenotypes. So we can actually just look directly at the mRNA target and see, has it been affected by this manipulation? So what we have with the zebra mussel genome, as I mentioned, is basically a catalog of all the genes. And one of the challenges that we have is that this is a very large catalog. So, the zebra mussel genome consists of almost 2 billion letters. If you were to print this out, this would be equivalent to about 600 copies of Tolstoy's War and Peace. And there are about 68,000 predicted genes. So this is a lot of information to analyze and to sort through. And as a starting point, we took some of those functional experiments that we did in the context of the Zebra Mussel Genome Project, where we looked at the expression of genes in the mantle, the tissue that makes the shell. We looked at gene expression in response to thermal stress, so basically heating them up to noxious temperatures. And then we were also able in the genome to assemble the complete collection, or what we think is the complete collection of all of the genes that are required to make the Bissell thread attachment fibers. So these are the fibers that they use to attach to surfaces, attach to boats, and are likely to be, or are very important in their dispersal mechanisms. So based on these initial experiments, we were able to come up with a prioritized list of about 150 or so candidate genes that we felt were expressed at high levels in the tissues that were in important tissues or involved in very important processes that would make potential targets for intervention and biocontrol. We also had a few additional criteria because 150 is already a lot to kind of screen through as we're getting started. So we had some additional criteria that we used to further winnow down that list and zero in on targets that we thought would have a really high probability of seeing a phenotypic effect in our initial tests. So one of those criteria was that we looked at genes that had conserved essential function. And so these are genes that are absolutely required for life, for survival across a wide variety of organisms. So we looked at genes that were required in mice, human cells, worms, flies, fish. And you know, with the hypothesis that these genes would also be very important and foundational for zebra mussels. So the first example that I'm showing here, this RPABC5 gene, this is an alignment of the protein sequence of this RPABC5 from flies, worms, mice, and zebra mussels. And what you can see is that this gene is almost identical across all these different species. And this is a bit of a rarity. So zebra mussels are pretty divergent from anything else that's been sequenced. And so we don't see a lot of this level of conservation. But you know, the fact that this is so conserved suggests that this is a very, very important protein. We know that it's from studies in other organisms that it's a component of the RNA polymerase II complex, which is a complex that's a protein complex that's required for expressing any gene from the genome. So it's, you know, it's involved in a very basic cell biological process. And so we thought that this would be a good target, you know, just as an initial test, because if we can really knock this gene out or knock this gene down, the animals really should not be able to survive that manipulation. We also looked at other gene targets, and you can see, you know, some of these are less conserved. So the second example here is a Shaker potassium channel. It's a gene that's required for nervous system function. So you know, we use that as that evolutionary conservation as a guidance for what genes are likely to be very important. We also looked at genes. We also looked for genes that were single copy. So oftentimes important genes or many genes will be duplicated or have multiple copies in the genome. So we wanted genes that were just a single copy so that we could know that we were manipulating just one target. We also wanted to find genes where we felt that there'd be a possibility of having a rapid assay, a rapid phenotypic assay for assessing the effects. So for our initial screening, we zeroed on genes that we felt would be required for viability or for surface attachment. And then because we also don't know what tissues are potentially susceptible to the introduction of these RNAi constructs, we wanted genes that were predicted to have effects in a wide variety of tissues, so we could sort of cast a wide net and screen through lots of possible options. So over the past 9 months or so, we've narrowed the list down to about 20 initial candidates, and these are genes that are either involved in highly conserved cell biological processes, you know, DNA replication or repair, you know, transcription, nervous system function, or genes that are, or this collection of genes that are involved in forming the Bissell thread attachment fiber. And we've been working on making RNAi constructs and now have RNAi constructs for pretty much or almost all of these genes. And this has been the work of Lindsay Engelbeck and Margaret Donovan in the MGC. So with that, I'm going to hand things over to Scott, and he'll tell you a little bit about the work that we've been doing in the containment lab to establish some of these phenotypic assays.
Okay, can you hear me now?
Yes, we can hear you.
Sorry about that. Yeah, so thanks, Cheryl. I want to update you on the progress we've made on the last two parts of the procedure. And I want to introduce myself. I'm Scott Ballantyne. I'm a professor at the University of Wisconsin River Falls, and I'm also a property owner on a lake that's infested with zebra mussels. So I'm very interested in seeing this project through. For our project to work, it's critical that zebra mussels eat bacteria, right? They've got to ingest these modified E. coli that Daryl described. And a few years ago, I did the experiment shown here together with some students at UWF. And what we did is we put small mussels into each of these plastic tubes. We then filled the tubes with water, and then we added E. coli to these tubes, but it wasn't normal E. coli. These were E. coli that had been genetically modified to glow green. And the first 3 tubes are experiment, so we left those alone. But the second 3 tubes were our control, and to those we added a powerful toxin called sodium azide. And initially the tubes glow this nice green color, but over time what we saw was that the tubes with the live mussels, our experimental tubes, the glowing went away. And in fact, by 4 days, it was basically gone. And again, importantly, the control still glowed. So we can conclude from this that zebra mussels can in fact filter E. coli, but it took a while, right? So we wanted to increase the uptake, and that's something we've been working on in the lab this summer. One reason we want to increase uptake is also shown in this paper. Daryl just mentioned that oysters can do RNAi, and the authors of this paper used the exact same system we're trying to use, right? They used engineered E. coli to both produce and deliver these double-stranded RNA. And what they found was that the E. coli didn't work all that well on their own, but they worked a lot better if they combined them with an algae. So, and what they think is going on is that the algae are the E. coli are sticking to the algae, and then the algae are delivering the E. coli to the gut of the oyster sort of as passengers. Right? So they came up with this term Trojan horse strategy to describe their procedure, and I think it is logical why they would use that. So we thought, hey, maybe we should mix our E. coli with algae. And that led to the question of which algae. And we're actually trying several, and I just want to tell you about one. The algae is called Chlamydomonas reinhardtii, and I'll probably just call it clammy from now on. It's a safe, harmless algae that's found naturally in our lakes and in our water, in our soil. It's relatively easy to grow, and there's some of the world's experts in working with this algae are present on the University of Minnesota campus. This is one, Pete Lviv, and he actually co-directs something called the CRC, which is the Chlamydomonas Resource Center. And Pete and the other staff at the CRC have been really helpful in getting us started working with this algae. And we now have nice green flasks of the algae growing at the containment lab. What gets me most excited though about this algae is that it can be genetically modified. So we can use the algae in the same way that we're using E. coli, at least in principle. We can create algae or clammy that are producing these double-stranded RNA. We've spent several reports recently using this strategy. In this first one, they generated clammy that was targeting a gene from a virus. And that virus is a virus that infects shrimp. And I guess they raise these shrimp in aquaculture facilities, and periodically they get die-offs because of infection by this virus. And what they were able to show, at least in a contained setting in tanks, was that if you fed the shrimp this modified algae and then later challenged them with this virus, they were much more resistant to infection. And so really what I'm describing then is a type of vaccine, right? This algae is acting as a vaccine, and it's preventing these shrimp from becoming infected. And that to me is a really kind of new, at least for me, it was a new way of thinking about genetic biocontrol. But I've always considered biocontrol as something we would think about using after a lake becomes infested. But maybe we should, at least with this algae type of system, be thinking about it as something you could use proactively, right? Something you could use to protect a lake from future infestation. In the second paper, they used the same strategy, but they targeted a gene from mosquitoes. And this is a gene that the mosquitoes need to develop. And again, what they were able to show, it was in a controlled setting, if they put that algae into water, the mosquitoes reproduce less well. And their goal is to try to limit mosquito-borne diseases like malaria. So I think this modified algae approach has a real future, has a real potential long-term as a way to effectively deliver these double-stranded RNAs in aquatic systems. So there's our strategy again, and clammy is really going to play 2 roles for us. The first role is it's going to act as our Trojan horse, right? It's going to help us deliver our modified E. coli into the zebra mussel gut. And the second idea, more long-term idea, is that Clammy is going to act as a substitute for the E. coli, actually produce the double-stranded RNA for us and produce them directly. So I made some progress with the second approach. I was on sabbatical last fall, and I was able to design a plasmid that we creatively called P-Clammy. And that plasmid is basically, it's based on the shrimp paper, but I tweaked it a little bit so that it's better able to receive our target gene. And Lindsay and Margaret went on to make the plasmid and show that it works. We put a few now target genes into this plasmid. And it's actually a project for my molecular biology course this semester to put this plasmid into Clammy. It turns out it's not a single step. It's actually fairly complicated, but hopefully we get that to work and we'll have some of those algae available to test next summer. Okay, with the time I have left, then I want to talk about what we're going to do once we feed these microbes to the zebra mussel. Right? What are we going to measure? What are we going to monitor? That's what we mean by phenotype. And I've listed several criteria there that I think would make a good phenotype, at least to initially test. We want something that's simple to score, and that's because we're not all zebra mussel experts. Something that's quantitative, so we can do statistics. Something that occurs quickly. That's because I'm not patient. And something that we have good gene targets for. I think that's obvious. And perhaps most importantly, we want something that's connected to muscle health. Right? As much as Daryl and I might want to change the stripes on these mussels into polka dots, that's probably not the best trait to focus on. So here I've listed some of the traits we've considered, and I broke them down according to whether they happen quickly or more slowly. And I'm only going to focus on the fast ones. Filtration is a trait that I've already shown you a little bit about. I'm not going to say much more about it. Movement, it turns out zebra mussels do move. I did some experiments early on looking at zebra mussel movement. The problem is that they don't, not many of them move in any given experiment, so there's not much signal. So I've kind of ruled that out as a good trait initially. And that leaves us then with attachment and copper sensitivity. Attachment is really important. Zebra mussels need to attach to their substrate in order to feed and to breed. So I came up with a rather simple test for attachment, and technically it's reattachment, that I made this graphic to describe. I jokingly call it zebra mussel Yahtzee. And the basic idea is you take mussels off their native substrate, in this case a rock, and you drop them into a jar that has water. You then let that jar incubate for some period of time. We usually go like 24 hours, and then you cap the jar. And this would be the point in Yahtzee where you shake the cup, but we don't shake the jar. What we do is we cap it and slowly invert it once, and then we dump it and count the number of mussels that are left attached to the jar. So this reattachment assay meets a lot of the criteria I listed earlier that we're looking for, at least in our initial phenotype. We want something that's simple to perform. Well, this is definitely simple to perform. Something that's quantitative. We get numbers out of this. Something that occurs quickly. We get reattachment in almost full reattachment, sometimes in 24 hours. And something that we have good gene targets for. And I didn't go into this, but the structure that attaches, the byssus, is one of the most studied structures in the zebra mussel. And we have a number of good gene targets that encode components of that structure. So that leaves us then with the question of, does this reattachment have anything to do with muscle health? Well, I realized one of the great things about preparing a talk like this is it gives you a chance to sort of step back from your day-to-day experiments and think about your data collectively. And I realized that sort of hidden in the data from this summer, we already had a partial answer to this question. Because what I'm showing here is the control reattachment we got for various Yahtzee games that we played throughout the summer. And so these are just our controls. And the point I want to make is that there's a fair amount of variation, right, in reattachment between the various experiments. So that could mean then that these different batches of mussels have different health. Some are more sick than others. But there's a problem, and that's that really wasn't the goal of these experiments. And so there's some other variables here. These were collected obviously at different times of the year. The mussels weren't always the same size. They're attached to different substrates. And so one of those other things could be contributing to this bounce. So I wanted to do a more controlled, careful experiment to really figure out if there's a connection to muscle health. So the experiment the design of the experiment is shown here. What I did is we collected mussels from a common source at a common time on a common substrate. So it was from Lake Minnetonka on rocks. And then I selected mussels that were approximately the same size, and I distributed them among jars. Now I wanted to make the mussels in some of these jars sick. And so I chose to do that by adding increasing amounts of a chemical called copper sulfate. I knew at high levels of copper sulfate that it would kill the mussels. So my thinking was low levels should make them ill. And then we did the reattachment assay. So here's the results from one of the trials. And what we saw is that with no copper, you get pretty good reattachment. With high levels of copper sulfate, you completely eliminate reattachment. But importantly, at these intermediate levels, you consistently, get intermediate attachment. We did these experiments several times, and we always saw the same thing. These low doses of copper always inhibited reattachment some, but not completely. So I was pretty excited by this result. To me, it says that reattachment is indeed a proxy for zebra mussel health. And the reason that's important is initially I thought this little test would be useful for testing those genes that are directly involved in attachment, like the Bissell genes. But this result, I think, argues that maybe it's going to be a test that could be useful in a broader way to test a lot of our genes that are involved indirectly in attachment, but directly perhaps in muscle health. So to think about how this could work, I made this slide. The basic idea is we would take our microbes that are targeting various genes, here gene A, B, and C, and we'd substitute them for the copper in the previous experiment. And so we'd put them in, and then we'd ask which one, what happens to reattachment in the presence of these various microbes. And so everything on the right here is hypothetical, but if we see gene A and gene C attaching about the same as control, we're not real happy. But if gene B it gives this reduced reattachment, essentially behaving like copper, that gets us excited. Now, the problem with presenting you a slide like this is hopefully you understand it, but it's really deceptive. You think we're going to have this project done next weekend. And there's a lot of things that need to be optimized here. We're currently working on the composition of the treatment, right? Do we include algae? Which algae? How much? The dosing, how much bacteria do we add? How often? How long do we go between treatments, and how frequently do we treat? So the devil's in the detail. So another reason I got really excited about the copper experiment is it indicated, to me at least, an immediate way that we might be able to use RNAi to make mussels more sensitive to copper. And the idea here would be we would treat the mussels ahead of time with genes, with microbes that are targeting genes that could be involved in copper resistance. And this isn't just hypothetical. We actually have a group of genes that we predict would be involved in copper resistance. So we knock down those genes, and then we repeat the copper test. And what we'd be looking for are, this would be the control, just what I showed you a moment ago. But if we have some hypothetical gene X that's involved in copper resistance, it should shift the curve to the right. Right? So it should make mussels that are in low doses of copper behave like they're in high doses of copper. And the reason that's interesting is because there's great work going on right now by Diane Waller and Angelique Dahlberg, where they're actually testing copper in infested lakes. Right? So they're talking about using this in low doses to treat infested waters. And we might be able to, with RNAi, make that work even better. By better, I mean more specifically in allowing them to use less copper. Okay, so I want to quickly end then by just listing 4 ways I could see RNAi contributing to zebra mussel management. I want to stress we're not going to get these all done in the next year. One way would be to identify gene targets that could be used for DNA-based biocontrol. This is the sort of work that Mike Smansky is doing, so CRISPR-based strategy. It could provide a real powerful experimental tool that helps us understand mussels at a whole new level. But I'm most interested in these last 2, the direct ones. At least in principle, RNAi could reveal new double-stranded RNA mullicides that would be unique in that they would only target zebra mussels, right? They'd be extremely specific. And finally, the holy grail, it may be possible to create a whole new level of RNAi-based genetic biocontrol agents. And the idea here would be to release like those modified algae, for example, into infested and perhaps even uninfested waters, And the advantage here is that, these are alive, so they replicate, make more of themselves, and they provide a continuous fitness defect on the muscle population. And that to me is the only real way to come up with a cure to these. I want to end them by thanking MACERC for supporting this, organizing the showcase, and for supporting us through the Environmental Natural Resources Trust Fund. I want to thank UW River Falls for supporting my sabbatical. And most of all, I want to thank Daryl Goyle, he's the project manager. He's an amazing scientist, an amazing guy. It's been a lot of fun to work with. So with that, we'll take questions. Oops.
Thank you so much, Daryl and Scott. For attendees, a reminder to drop your questions into the Q&A box. To kick us off, I think Daryl and Scott, if you could, you touched on this in your presentations, but just to kind of directly ask the question about how this RNAi technology would be applied through either the algae, or would it be through an introduction of a mussel that it would then breed into a population? Can you just kind of touch on that again, about how down the road would these genes be released into a population?
I guess I'll take a shot. Do you want to, well, the one way I see as viable and quick would be to actually make the double-stranded RNA. We have a way to do that synthetically, or you could actually just grow up large vats of either the E. coli or the algae, and then kill everything in it, and then just release that. So it's basically you're providing the double-stranded RNA in a similar way to thinking about how they're providing copper. The problem I see with that is we have no idea if we could provide enough, and it's almost certainly going to require multiple treatments. It's not a cure, right? It's a treatment. So longer term, I would much more prefer to see the double-stranded RNA delivered through something alive, like the algae, because that has the potential to make more of itself and so provide a continuing effect. In other words, I don't want to use the word cure, but closer to a cure.
Yeah. Our next question is, have you done any studies on how or if the biocontrol is affecting native mussel species?
No, we haven't looked at that yet. So we do actually have a large collection of DNA in our freezer now that we've gotten from a variety of native mussels and also fish and other organisms. So, that's definitely an important question to ask before one would contemplate, doing something actually out in a lake. And, I think we can potentially look at that specificity. Right now, we're really focusing on trying to see, is this approach viable, from first principles, right? Can we see any phenotype in zebra mussels that indicates that RNAi would be an effective tool? I think once we've answered that question, then the question of specificity and becomes a lot more salient.
And Daryl, you touched on this in your presentation, but again, for clarification for attendees, you talked about kind of how broad, or not broad, but how massive the zebra mussel genome is. How do you go about, once you have mapped it out, identifying what all of the different genes do? Can you give a quick summary of that?
Yeah. So I mean, there's a lot of this is done computationally, right? So we can't sit down and look through those sequences by eye or by hand, or at least not for a large number of them. There actually were an interesting class of genes that were involved in shell formation that did stick out by eye. They had these like long stretches of these GGY repeats in their structure, and so like when you look at them, you say, wow, that's like, there's something weird about these, like, very highly expressed genes. But that's sort of the very small minority of cases. So generally what was done is that we used a variety of tools that allow us to computationally predict genes. And then we refine those, and that's based on homology with other organisms. So I showed you some examples of conserved genes and how you can use kind of that alignment with different organisms, and then knowledge about those different systems and the functions of those genes to annotate the genome. So we use a combination of that evolutionary or kind of sequence relatedness between different organisms and homology to computationally identify genes. Those predictions were also refined by some of the gene expression data we had. So obviously your confidence in those gene models grows if you see that gene actually expressed in the mantle or in the gill or in different tissues, right?
Mm-hmm.
Right now we have that list of about 68,000 candidate genes. I think that that's probably an overestimate of the number of actual genes that are in the zebra mussel genome. And this was also seen in the human genome, where initial predictions were over 100,000 genes. And now I think with 20 years of work and refinement of that list and experimental testing, it's now about 25,000 or so genes in the human genome.
And can you give us a sense of kind of the timeline for developing technologies like this? I mean, how are we like next year, 5 years, 10 years? Is it hard to say? Kind of that horizon on something like this being implemented?
Yeah, well, so, I think right now, what we're hoping, throughout the course of this 2-year project is to answer that question of whether RNAi is a viable approach for making genetic manipulations in zebra mussels, right? So that's really, at this point, sort of an academic or research question, right? If the answer to that question is yes, then, then it opens up the possibility of doing the types of things that Scott discussed. Obviously, those are going to be contingent upon, further technological development, right? Of like refining those potential delivery mechanisms. And then also the types of conversations that, David Fulton and his group have been starting to initiate with their MESARC project of engaging with the public and trying to kind of craft a plan for safety and the sort of political and societal considerations of employing these tools, right? I think the good news there is that there are a number of systems where these types of approaches and tools are much more advanced than they are for zebra mussels right now, right? And so I'm thinking about like mosquitoes, where genetic biocontrol is already being tested in the field, right? So I think, for better or worse, zebra mussels won't be the guinea pigs in the initial test bed for deploying these types of things in the wild. And, we'll be able to learn from and see the experiences of these other groups in thinking about and making those longer term decisions.
That's a helpful framing. Thank you. So this question is somewhat related, but touching on kind of those ethical concerns that you talked about and the interface with the public and kind of the viability when it comes to the social side of these kind of technologies. And the question is, zebra mussels got here, so there's a possibility that this algae could accidentally get somewhere else. How is that addressed in the technology or in the research? And is it being, it's not, if it's not a part of your project now, kind of how is that addressed in the process?
Yeah, so I mean, I think that's that's definitely an astute observation, that the downside of what Scott discussed of releasing a living genetically modified organism into the wild is that you then, how do you control it, right? Or how do you limit its spread, or how do you make sure that it doesn't have off-target effects that you can't anticipate, right? And yeah, I think that that's a really important thing to consider before ever employing something like this, right? There are sort of experimentally, people have developed like genetic kill switches and things like that. So you could engineer certain certain susceptibilities into that organism that you're releasing, so that you could potentially control it. But there's also evolution, right? I mean, we've all seen Jurassic Park. We all know that those types of things might not be a a complete, fail safe, right? And so I think it's definitely a really important point to be bringing up and to be discussing, and something that will need to be dealt with for any sort of like field application.
Thank you. It is 12 o'clock, but we did have one more question that I think is a good one. So if it's all right, I'm going to ask one last one, and then we'll wrap up. And the last question is, as copper is a broad toxicant to many aquatic organisms, and its broad use in the natural environment is complicated, what pathway is the next best strategy the lab is examining in terms of a potential genetic biocontrol, i.e. interference with shell formation, thistle creation, or waste production?
Well, we have, like I showed, we have good assays for filtration, and I think that would fall under that category. I think I might have confused the questioner though, because we're not actually, the copper targeting is one small subset of what we're hoping to look at. Reattachment is not just about copper. It's actually mostly not about copper.
Thanks for that clarification. All right, I think that wraps up our questions. If you have questions as attendees that pop up as you're kind of thinking about this presentation over lunch, feel free to reach out to Daryl or Scott. Their contact information was up on the screen, or you can reach out to macerc@umn.edu. All right, thank you, Daryl and Scott. We do have a few minutes for questions. So if you have questions, again, either points of clarification or kind of expanding on the presentations here, please drop those in the Q&A box. I can kind of kick us off with lots of times when we talk about this project, both when the mapping of the genome was happening a few years ago and now, we get the question of how is this technique, this approach, how do you know that it's species specific and won't affect native mussels or other kinds of mussels in water bodies once it is released? Can you kind of speak to that? You touched on it a little already in your presentations, but a little more directly answer that question.
I'll try. It's sort of a two-level answer. The first is computational. We do our best to identify a DNA sequence that's only found in zebra mussels, but recognize we haven't sequenced everything. So we'd look at all the things we'd sequenced. We've sequenced bluegill, walleye, stuff like that, and we'd look through all their genomes and make sure that whatever sequence we're considering using doesn't accidentally line up with one of their genes. But the problem is we haven't sequenced everything. So it will also take studies by scientists, not like myself and Daryl, but more traditional organismal type biologists to go out and sample these in sort of a controlled environmental setting and make sure they don't do collateral damage that we're not thinking of.
Thank you. Another question that came in is, what are the potential negative side effects of the algae approach that you presented on, Scott?
Yeah, it's a pretty fresh idea. So what else? Let's spin that. Let's talk about the positives. The positive of it is you're not adding an invasive species, right? This creature is already in our lakes. So in fact, I asked that question of Mike Smansky just a moment ago. He's talking about using his carp prophylactically, but those are invasive. So our algae would already be in the lake. So the danger, if there is one, would come from what we've added to the algae. Right? And it would be this little bit of DNA that is making these small double-stranded RNAs. There's strategies, of course, to control the algae, just like any modified creature, which could fail. But if it failed, you'd have an algae in the lake that's already there.
Okay, that kind of addresses another question that came in, but if this technology works, after zebra mussels are eliminated from the lake, what then happens to that special algae? Does it stay in the lake? Do you what's that next step? Can you address that a little bit?
I could try. you know, I tend to not, I'm not thinking that far ahead. I'd love to see it work in
Yeah.
the lab, and I'll just stress that we're working only in the lab. But it's worth thinking long-term. And, you know, like, as I mentioned, I know there are tools that we can engineer into the algae that would make it self-limiting. And one of the great things about having this CRC resource right nearby is they have every mutant algae ever created right there. So I would right away go to Pete and say, how do we do this? How do we keep these algae from being able to perpetuate indefinitely? You know, how can we build in a safety net?
That's great. With either of these kind of, and I guess jointly this approach, assuming that it works, this is kind of a hard question to answer, but can you project out kind of what the cost would be for this management tool? Would it be something that is very expensive, only to be used in kind of key places, something that's accessible? Kind of speak to that timeline a little bit.
Yeah, I think it really depends on the pathway that is taken to apply it in the field. So, you know, Scott mentioned there could be a few different ways of actually doing the application, right? One would be essentially a chemical approach, which would be, you know, producing these double-stranded RNAs and applying them. That would be, you know, a local application. You could also grow up large quantities of these bacteria and kind of heat kill them, and then, you know, and introduce the genetically modified organisms that aren't capable of reproducing in the lake, right? And actually, there's a precedent for that already in treating zebra mussels, which is Zequinox. That's actually derived, or it's a bacterial treatment, right? So it's a particular strain of Pseudomonas fluorescens that causes the guts of zebra mussels to deteriorate, and it's introduced in, you know, large quantities. That approach, I mean, the problems with that is that it is expensive and it's difficult to, you know, it can only be applied in sort of local areas, right? So, you know, I think short of introducing these kind of live organisms to the wild, RNAi as a direct strategy would be likely to be probably on par with Zequinox. It probably have similar limitations in terms of its both expense and effectiveness. But, you know, I think right now I see a huge value for this work in, you know, giving us a research tool, giving us a way to potentially manipulate and ask questions in zebra mussels, laying the groundwork for a future when we could apply, you know, DNA-based tools like CRISPR for population control in a way that's similar to you know, things that are being done in mosquitoes and you know, other undesirable organisms right now. And, you know, I think that there is a potential for this approach to be directly applied in the ways that, you know, that Scott mentioned. But, you know, that's obviously something that's going to require you know, that sort of public political conversation of, you know, what is the cost benefits of this. And I mean, I'm really glad to see that MASERC is starting to engage with people and start to, you know, get some information through the project that David Fulton is running, and, you know, how the public does perceive these types of interventions and does perceive those cost benefit calculations.
Okay. So in our last minute, I want to ask this last question that we got in, because I think it's a great kind of close for this session. But what do you think is the overall importance of this research, and how do you see it affecting science in the future? So kind of big picture, how is this making a splash in the field?
Well, I can speak for myself, and maybe I'll let Scott take a crack too. I mean, for me, you know, this is a really challenging problem, right? So I'm trained as I trained and did my PhD in fruit fly genetics, which is a really, you know, well-established genetic model system. It's got 100 years of technology development and tools that make it very easy to kind of make these types of manipulations. Zebra mussels are complete uncharted territory at this point, right? So just learning the basics of how to work with them and how to, you know, keep them alive, how to test things, and, you know, develop these assays that we can use to study phenotypes. You know, I think that's really laying a lot of groundwork for, you know, being able to do more sophisticated things, right? So, you know, I think hopefully at the end of this 2-year project, we'll have some indication of whether RNAi is a viable approach. And that's going to either give us a very powerful research tool or, potentially rule this out as a strategy and cause us to look in a different direction.
I see we're out of time, so I'll just say I basically echo his answer, and I tell my students it's the ultimate final exam for me because this is the biggest scientific puzzle I've certainly ever tackled. These things are tough to work on. It seems like everything's moving. So just little advances, right? Figuring out how to effectively deliver E. coli to them. Yeah, that's a move forward. And science moves in small steps. And so even if we don't solve the problem, we're going to make a contribution.
Thank you both so much for your work and for presenting for us today. I'm going to wrap us up there.